Doing this, then we go to the definitions we need. Right? This is just these 2 slides for technicalities. The strategies I'm using for building concepts, there are 2 here. One I already told you, starting from concrete objects, which is living beings, and then proceed to abstract concept, which is life. Yeah? And when we are studying these concrete objects, we start from the outside, which is the interactions they make with us. We are the observers, and the interactions they make with us is through the phenotypes, through the activities that we know. And then we proceed to the inside, to the things that are there in the middle of the cell, in the kernel, in the core of the cells, that we need analytical procedures to get there. Yeah? Then from the outside, we can say that living beings are cells, living beings are metabolic flow systems that take things from the environment, transform in the inside to build their own things, and excrete, and make a flow system. Something enters, and then others go out. Then there are metabolic self-systems, metabolic flow. And in the inside, we have these 2 things. The metabolic flow allows the cell to construct itself on the basis of memories. The memories are that part, the left part of the halter there, which is RNA and DNA things that make proteins. Then we need that memory so that we can construct ourselves. Otherwise, we would have to invent ourselves all the time. And the other part is that they adapt and evolve on the basis of plasticity. And this plasticity is built in the stuffs that we are made of. Most plastic is protein, less plastic is DNA, intermediate is RNA. And it is on the basis of this plasticity that we can adapt and evolve. If we are not plastic, we don't adapt or don't evolve. This self-construction is something that is being done all the time until now. We are self-constructing and reconstructing, regenerating, redoing everything we need every time. Then it's continuous self-construction and reconstruction. Memories, we have many. There are the memories in genes and memories on the metabolic pathways, memories in the interaction of the systems, which has some stable parts and some unstable parts. The stable parts of the metabolic system makes the epigenetic kinds of memories. And the plasticity is also not something miraculous that happened to be at the origin of the living thing. It was selected along the process of living. The basis of the selection is stability. What is, even before Darwinian mechanisms, we are not treating about Darwinian mechanism because there's no reproduction yet. The reproduction comes in the next slide. Here is just stability. What is stable stays more and will participate more in the next step. Then stability was the driver of selection of the constituents, but the problem comes when you are living on environments that are very variable. Then plasticity is needed, and this is a property of proteins and of the networks of metabolism that it does. This concept here can be then summarized in some keywords. We are metabolic systems together with the environment that we take things from, and then we give things back to the environment. We are autopoietic, which is self-constructive, while we are mnemonic. Mnemonic means we have memories. Mnemosine is the Greek goddess of memories. Yeah? And we are evolutionary because we are plastic. I took these three characters here and have to give lots of thinking to it, and I cannot stay with any one of them isolately. I think we need all three together. There's no way to have only memories like a replicator. There's no way to have only proteins that don't have the ability to replicate. We don't have any way staying with metabolism from the outside, which is geochemistry. We have our own metabolism, otherwise we don't survive. And then we need all these three parts. None of them, all of them are necessary, and none of them alone is sufficient. And this is a simple description of a living being, and life is the process that these living beings depict and construct along evolution, along ontogenesis, along their living times. Yeah? Then life becomes somewhat easier to define, and at the same time, it's not defined as an entity. It's a process. Life is everything we do. And everybody that tries to define life through one specific aspect will not satisfy the other, because there are many, many aspects. And these may change along your ontogenesis and along the lineages and populations that come around. Yeah? Life for a worm is different from our life. Yeah? There are some things in common, some things not. And here we have then a little picture of that. We can divide the living system into two parts. One part is metabolism with the environment, and the other part is the core, which is nuclear proteins. Metabolism is what sustains. Metabolism is what interacts with the environment, takes things from it, transforms into things that can be used to build the core and make things out. With this process of metabolism being transformer, it's obligatorily degrading the environment. Then we are at the same time autopoietic and basuropoietic. We are throwing trash into the environment all the time. And this is essential to us. We only know how to do it well. No? But this is necessary. There's no way to live without giving waste to the environment and taking things out from it. And this is very strongly noticeable because we need to know how to do it well. Otherwise, we will saturate and exhaust. We will exhaust from things that we need to take in and saturate with things that we think it would give back to it. The other is the internal. The left part here is memories, which is conservativeness, which is based on replication on the nucleic acid. And the other part is the proteins, which make the cells function. Everything that functions in organisms is made by proteins. There's no way to have things done by DNA, things done by RNA. Only very little RNA participates in metabolism, but the bulk of it is made by proteins. And proteins makes structures, functions, and it's flexible, and it's diverse. And metabolism is made by proteins also, a little bit by RNA, but most of it is protein. And this has a structure of networks. The network is very difficult to go along with. It's kind of a labyrinth. You don't know how to get one from the other. Then it is the flow that organizes these things. When we have these two things together, this is also what defines metabolism, because metabolism has to serve those two things through utilization of the environment. Then it's kind of a triangle organization. When we have these two things, they have it always in coevolution. There's no gene without a protein. You can only define a gene if you have the protein it makes. And you only have a protein having a gene that codes for it, then these things have to go together. It's always coevolution. The system, I already said that I consider a cell a protein production system. There's some machines there, which is a ribosome, and everything else serves this. The secret of being kept alive is to having a metabolism and protein synthesis. Metabolism serves protein synthesis. This is the main aspect of things. In the studies of life along the last century, that was a quest for looking of what would be the force inside organisms that make them go forward. This is mainly evidenced by the metabolism itself, because we are always looking for food, which is our main activities. And from bacteria to any kind, the main activity is looking for food. Then we'll have another one, which is reproduction, but that comes in later. The first is looking for food. Interact with the environment, take things from it, modify, and then produce the things from the core. And this core has the protein which is making those things that takes things out from the environment. Then this makes a circle. You have nutrition to make this, and this is make the proteins that can facilitate the nutrition. This is another circular organization. But we have one part of it which is directional, is not circular. One part that is direction is metabolism itself. It has the compartments for uptake, for transformation, and for excretion. This is maybe not linear, but is directional. There's a coming from the outside to the inside, and going then from the inside to the outside, excretion. This is easy when we have some micromolecules whose constituents are not nitrogenated. Then you say lipids, sugars, and things of this kind will be micromolecules, easy to excrete. We breathe out the CO2 and water, which are products of the transformations of all lipids and carbohydrates. And that's easy. It goes out with water. It evaporates. The problem comes when we have the nitrogenated components. That's the nitrogen problem. All the products of degradation of nucleic acids and of proteins will be toxic to some extent. Urea needs water to be dissolved with to go out with urine. The next component is uric acid. It's insoluble. Then it's complicated to get rid of uric acid. You have to transform it. And when you are making the macromolecules, which are nucleic acid or proteins, some of them are difficult to degrade and will stay inside the cell. Being difficult to degrade means that either they accumulate in a non-toxic form, but anyway they take space from the cell. And the other is that they may accumulate being themselves toxic to the cells. And the way the cells invented to get rid of them is to make vesicles and to expel the vesicles. Nowadays these things are called exosomes, extracellular vesicles, which are in medicine considered a lot as communication communication artifacts. That the cell expels some vesicles to communicate with other cells through these vesicles. But in the origin, these vesicles were only to expel things from the cells that, when these things were not apt for degradation. And they are proteins that cannot be degraded easily. They accumulate. Then they are packed into vesicles and go out. And the origin of reproduction is linked to these vesicles that are being expelled. Some of the vesicles acquired some memories inside them, and this vesicle with memories inside becomes a daughter cell. That's the origin of reproduction. And reproduction then is a part of the excretion system. Then we have two kinds of excretion. One is from macromolecules that takes out things that can be gases or transpired in vapor, or the others that cannot become degraded to macromolecules. They'll be expelled as chunks, as vesicles. And reproduction, funnily, it's a derivation from this kind of expelling vesicles, when vesicles acquire genomes. And then there are two parallels here that I like to do. One is with ethology and psychology that try to make which are the pulsions, that's the élan vital, that's the vital force and these things that organisms are subjected to. One is the nutritional, at the beginning of the metabolic flow, and the other is the reproductive, which is at the end of the metabolic flow. The cells, when they are not reproducing, they are expelling vesicles, and reproduction is one of those kinds. And this also solved one of the problems that I had. I remember my youthful times when that thing that intrigued me a lot of having the reproductive organs together with the excretion organs. Yeah? This, in animals, this is it's very calls the attention for that. And when we get to this thing of the metabolic flow, including the reproduction thing together with excretion, then this became explained. It's original. From the beginning it was that way, it was kept that way until now. The urogenital association, which is a complicated thing. Here are examples of how the vesicles are together with reproduction, and also from the physical-chemical origins of that. At the times we were studying origins of life, you would remember the coacervates of oparin, the microspheres of Sidney Fox. They make structures that look like reproduction. A mother cell producing gemmules and sprouts and buds, or growing from these buds coalescing to the big one, which look very much like the vesicles that come out of bacteria. Then this is physical-chemical, and this is biological, and they look very much the same. This is what started the whole thing with oparin and these people. An eukaryotic cell, which is very big, this is very large, has lots of those vesicles being put out. The reason for that is that the vesicles are being extruded to alleviate degradation, to benefit the general flow. The main part of the general flow is protein synthesis. These things would be lost, entirely lost to the environment, unless they got the memory to become daughter cells. This is just one of the examples of a cell in getting involved in reproduction. The first thing is to throw out something that would be a vesicle, a chunk of the cytoplasm being thrown out. Then it receives a genome, and then they become two cells. And the idea is that. This is a general conclusion from the metabolic flow studies. Everything that's in red here is metabolism. Then it is directional. We can model it as a crane. Here is dentate tape. Every time that dentate tape makes one step forward, which is taking up things in the uptake direction, yeah? Every time it makes one step forward, it moves the protein segment that goes together with that because it's taking up things. It moves that. You have to make more protein to keep that flow going. When the protein synthesis component is at work, it will move then the third part of it, which is the memories, and then you have to look for the genomes for what kinds of pieces are needed to restore and regenerate that will make the crane keep going. We may at some times try to model that thing also. Then at entry of 1 unit of substrate, then there's a coupled gear which is composed of 2 parts, the proteins, which is consumptive, regenerative, and the third part is to consult the memories of how we can get more of those kinds of proteins that are needed at that time. We may try to model that at some time later. When we have finished with the individuals, now we are coming to the populations. Populations live together, organisms, based on communication tools to have those populations working well. We are here treating some of the categories of communication artifacts that we use to build integrated populations. We are not treating here of reproduction. We only have taken, because reproduction is something individual, but it also involves populations when we have sexes, yeah? When we have genders working on the population. One important thing is that some people would like to take some of these categories here, like RNAs or DNA viruses, RNA viruses, as living beings. One statement that I used to say is that there's no life diffuse in space or in single molecules. Life is something, a property of systems that has different kinds of molecules doing different jobs together integratedly. The viral part of it is very interesting. You can have viruses of diverse kinds, from chemical elements like DNA or RNA or proteins, or some others that are typical of human communication. Yeah? Some of them are viruses, some cultural memes, some algorithmic viruses of computers, and they all would belong to the same category. Now the 2 last slides talking about some human type things. We can call diseases or as perturbations of the flow. This is something I would like to call medical people to work on, to transform those International Classification of Diseases, ICD, on divided into categories of different ways of perturbations of the flow. But what we are most interested is what came up as novelty on Earth with the flourishing of life. This is then consequences of the life process that were brought to the Earth. Looking at the organism, we see that there are no new forms of energy. We have only new types of organization and information being developed with organisms. There is this long time questions of organisms being dependent on accumulation of information, accumulation of organization, which is kind of Sisyphic type. Sisyphus is the Greek god for that we are always building organisms, increasing organization, increasing information. This looks like anti-entropic, but it lasts very little. We die after some years. We do it at about 100 years. Few organisms live longer, but it's always pseudo anti-entropic because it lasts little. The only attribute that the living process has that makes it really anti-entropic is considering the lineages and the transfers of organisms from one to the other of the information they had. It all goes through gametes and through culture. Yeah? But this is, it would be like the relay proofs in athletics. Yeah? One passes through the other, one passes through the other. Then what remains is population, is not individuals. We humans tend to be individualistic a lot, but it's kind of a specific type of attitude, because we know we last little, and these things has to go through population, through collectivities. I think this has implications a lot for the discussion of individualist and socialist type of attitudes. The human mode, every species will do it in its own way. I think an ant has some kind of conscience which is proper to it. A worm also does. The human mode is perplexed. Both these two here that I chose as representative are perplexed organisms. One perplexed in thinking about it, and the other perplexed with his poetry and looking at outside of organisms, outside of the earth. And we come to this situation that we are living now, which is the main worry. The living systems produced a lot of good things for themselves. We like to live, we don't want to die, but also we can do things that will harm ourselves. And this is taking to the extreme that metabolic activity, which recently was transformed into industrial activities. The industrial activities reach levels that surpass a lot the regenerative capacity of the earth. They are very intensive, extensive, devastating, and burners of everything. We are reversing that great event of oxidation that had happened along evolution before humankind came in. But when we came in and we entered this industrial area, we are doing, it's almost the reverse of the great oxidative event. We are now giving back all the carbon that we took in building ourselves. We are giving it back to the atmosphere, and we are reaching possibly reaching the limit. I think this is all I had prepared. I don't know if I extrapolated the time we had available, but it's all right. I hope you enjoyed it. Thank you.